Welcome viewers, welcome to the new lesson from Dr. R's Biology. If you did not subscribe the channel, you can just click on the subscribe button and bell icon. Welcome students. We have so far learned about the different classes of enzyme on the two classes we have already discussed. Okay, and the remaining four classes we are discussing in this particular lesson. Okay, hope you are already watched that particular lesson. If you did not watch that particular lesson, you have to first watch that particular lesson, part A of unit 2, topic 1. Then only you have to come here. Okay, so you have to watch part A first, and then only you have to watch the part B, that is this particular video. Otherwise you will get confused. So let us move to the third class of enzyme, that is nothing but the H, that is hydrolase. Okay, hydrolase enzyme. So hydrolase is the third class of enzyme. Now what is actually mean by the hydrolase? From the word itself, hydro is actually representing water. So it's doing something with water. Okay, so these particular hydrolase enzymes are nothing but the one which break the molecule by the addition of water. Okay, the molecule which break, the enzyme which break the molecule by the addition of water is known as the hydrolase enzyme. Let us see how it will be working. Okay, so you are familiar with a lot of enzymes in your body which will be digesting numerous macromolecules to its simpler compounds, simpler components, and that can be absorbed. For example, if you are just eating the glycogen, it will be just breaking down into glucose unit, and the glucose can be absorbed. When you are eating protein, it will be breaking down into amino acids, and the amino acids will be absorbed. When you are eating for example, fat, it will be just breaking down into fatty acid and glycerol. Then fatty acid will be converted into different compounds, and that can be absorbed. So that mechanism is actually with the help of enzyme which belong to the class hydrolysis. So I'm just taking the breakdown of the example that you are learning only. Okay, let us take the case of protein. Let us take the case of protein. So I'm just representing protein, the 2 amino acid part only, and let's see how this particular enzymes will be working. So we can see that an amino acid will be composed of an alpha carbon where it is linked with a hydrogen, an R group, and then the amino group. Okay, and then it will be with a C double bond O, and this particular link will be coming. I'm just adding the link with the red, the bond with the red, and this will be added to the next amino acid amino terminal. So N H, and then alpha carbon, hydrogen, so it will be the opposite direction. So R, and then hydrogen, and then again the C double bond O, O minus. I'm just representing a peptide bond. O minus, we can just say OH. It will be hydrolyzed to form the O minus. Okay. So this is actually the peptide bond that you can see in case of the protein. So this is the peptide bond. And the molecule that is represented here is a dipeptide. Because 2 amino acids are linked through a single peptide bond, and as a result we can see that it is a dipeptide. Okay, so we can see in order to break the protein into the simpler molecule, that is amino acid, that can be absorbed in your digestive tract, we have to break the peptide bond. So let us see how this peptide bond will be broken by the hydrolysis enzyme. So in this we can see that a water molecule will be added. So I am just drawing it like this because we just want to represent the entire thing. I don't have space there. So what will be added? Water molecule will be added. So I am just adding water molecule into it, H2O. So let us see where this H2O will be going. So this H2O is nothing but HOH. Have to keep in mind because you have already learned about the molecular structure of water. So we can see here this particular OH will be added at the carboxyl end and hydrogen will be added at the amino end. Then amino end is nothing but where you can find the amino group, and carboxyl end is nothing but where you can find the carboxyl group. You will be finding that in protein. You can just see the chapter protein lesson. So what will happen? Let's see. So one amino acid, it's one. So NH2. Then alpha carbon, hydrogen, the side chain, and then C double bond O. And what will happen to here? Here the OH from the water will be added. OH. Now the second amino acid plus what will be added at this particular point? It will be nothing but the hydrogen. So hydrogen, and then you can find this particular nitrogen, hydrogen, carbon, hydrogen, side chain, and C double bond O, OH. So you can see two amino acids. This R will be varying among different amino acids. If it is glycine, it will be H, just like that. So this is the reaction that is catalyzed by the hydrolysis. Which enzyme it is? It is actually peptidase, or you can just say pepsin, trypsin, chymotrypsin, etc. are the example for this hydrolase. Even your salivary amylase, which actually break carbohydrate into the sugar glucose units, it's also a hydrolase enzyme. The lipase, which break the lipid, also belongs to the hydrolase enzyme. Now let us move to the fourth class, that is lyase enzyme. As I told you, remember the classes of enzyme in that order only because the number is actually a matter. Because the first digit is actually representing the 4-digit number. Okay, so let us see the fourth one. Now the fourth class of enzyme is nothing but the lyase. So the fourth one is lyase. Lyase, we can just see that it is having something which is related to lysis. We have learned about hydrolysis, breaking, lysing, with addition of water, hydrolysis. The same similar kind of meaning is coming from the lyase. This is also doing that kind of thing. Non-hydrolytic cleavage of molecule is done by the lyase enzyme. Okay, so lyase enzyme is nothing but which are catalyzing reactions in the way that there will be non-hydrolytic cleavage of molecule. The cleavage of molecule without addition of water, without the utilization of water. That will be done by this particular class of enzyme. So non-hydrolytic cleavage. So non-hydrolytic cleavage of molecule is done by the class of enzyme known as the lyase. When it is actually performing the non-hydrolytic cleavage of functional group, we can see that there will be an insertion of double bond. Or if it is in the reversible fashion, if it is adding a functional group, then there will be the removal of a double bond. So in this way, it is actually responsible for addition of a double bond. and removal of double bond. So it is responsible for addition of double bond as well as removal of double bond. How it is responsible for addition of double bond? You can see that it is by the removal of atoms or groups. You can see that when a functional group is removed from a molecule, there will be addition of a double bond in between that. Let us see the example and you will be familiar with that. Now, how the removal of double bond? The same opposite way. Addition of atom or molecule or group. It will be responsible for the removal of. So let us define Lyase. Lyase are the class of enzyme which catalyzes the reaction which is responsible for the non-hydrolytic cleavage of molecule with addition or removal of double bond by removal or addition of atoms or molecules. That is what actually the definition for Lyase. Just rewind the video and learn the definition once more. Now let us see the example. I am just taking an example which you will be learning in Krebs cycle. Okay, in Krebs cycle there is a step where fumarate, that means fumaric acid, is converted into malic acid. So fumaric acid is converted into malic acid, or the malic acid is converted back to fumaric acid. Let us see that step, and that is the best example for the Lyase class of enzyme. The enzyme name is nothing but fumarase. So let us see that particular example. So fumarate is converted to malate by the addition of water, and the malate is converted back to fumarate by the removal of water. Who is responsible for this reaction? It is catalyzed by the enzyme known as fumarase. It is catalyzed by the enzyme known as fumarase. Okay, so we can see here. Let us see the structure and how it will be working, whether double bond is where and there. So in order to understand whether double bond is added or removed, you have to see the structure. So as the Krebs cycle and their molecule structure you have to learn. We can just take the example from the Krebs cycle itself so that you will be understanding what is actually the Lyase enzyme and how it is performing this particular reaction. So Lyase is a dicarboxylic, dicarboxylic acid. So and it is composed of 4 carbon. And there will be a double bond between the carbon number 2 and 3. And at the both end you will have a carboxylic acid group, and there will be 1 hydrogen here and 1 hydrogen here. Okay, so let us see how this particular reaction is happening. So this is what actually mean by the fumarate. I'm just representing the reversible reaction, and we can see that water molecule is added. It will be added to form the malate. So malate is also having 4 carbon and it is also a dicarboxylic acid. And you can see here COO minus, then C, then C, then COO minus. We can see that already hydrogen is here and hydrogen is here. Okay, so hydrogen there and here. So let us see what is happening there. We know that double bond is here, there is no double bond there. So this double bond is removed by the addition of water, where hydrogen will be added and the hydroxyl group will be added here. Okay, so this is what actually this particular malate is. So you can see that due to addition of water, double bond is removed. Due to removal of water, double bond is added. and it is removing water non-hydrolytically. It is adding water, okay, without forming anything. You can see that is example for the Lyase class of enzyme and its mode of action. When you are seeing this water here, you don't relate with the hydrolysis and all. Hydrolysis is the breaking with addition of water. Here there is no breaking of molecule by the addition of water. Instead, a double bond is removed by the addition of water. It can be any group, not only water. As you have to study this particular example in Krebs cycle, I am just taking this. Okay, so hope you got what is actually meant by the Lyase. Now let us move to the fifth class of enzyme. So the fifth class of enzyme is nothing but the I, which is actually isomerase. Isomerase enzyme. So you can see here isomerase. We know about isomer. Isomerase is nothing but isomer forming. Okay, so isomerase is an enzyme which catalyzes a reaction in which isomers of substrates are formed. Okay, so it is resulting in the production of isomers of a particular substrate. So you have a lot of example that is there in case of metabolism that you are learning, glycolysis, Krebs cycle, etc. And from that on itself you can just take an example. Okay, so what is mean by isomer? If you are not remembering what is mean by, so this is actually catalyzing the production of isomer formation. What is actually mean by isomer, that you should be familiar? The molecules which are having same chemical formula but different configuration. Now what is mean by configuration? Configuration is nothing but the spatial organization of atoms in a molecule. Okay, so it's having same molecular formula but different configuration. Now what is meant by configuration? The spatial organization of atoms in a molecule. Okay, for example, take the case of glyceraldehyde that you have learned in this carbohydrate part. Glyceraldehyde is having formula CH6O3. Okay, so this is actually the formula. And dihydroxyacetone is also having the same formula C3H6O3. But they are isomers. How their spatial organization is different. How it is different? In case of the glyceraldehyde, we can see that it is CHO, CHOH, CH2OH. The first carbon have oxygen and hydrogen. So let us see the dihydroxyacetone. It will be having CH2OH. 2 hydrogen is added there. Configuration is different. And second carbon C double bond O is having CHOH. And the third carbon is same. So dihydroxyacetone and glyceraldehyde are isomers only, because one is keto and one is aldo sugar. But still the chemical formula is same. So that is what actually mean by the isomer. And the isomerase are the enzyme which catalyzes the reaction which convert one substrate into its isomers. Okay, so this particular enzyme which convert glyceraldehyde to dihydroxyacetone or dihydroxyacetone to glyceraldehyde is nothing but triose isomerase, because the triose sugar isomers are formed. So let us see another example for this particular thing. So take the case of what we can say. So glucose and fructose are there. So the same thing. Glucose and fructose have the same formula, C6H12O6. So glucose and fructose have the same formula. We can see that they can be interconverted by isomerase. Glucofructoisomerase can be there. Okay, so you can see here one of the step in glycolysis is same only. Glucose 6-phosphate is converted into fructose 6-phosphate. You can just take that example. The formula will not be like this. It will be just different because the water molecule will be OH will be removed and the OPO3 will be added. Anyway, you can just see both have the same formula, but it will be having different configuration. Glucose 6-phosphate I am drawing first. CH2OPO3 2 minus HOH HOH OH HOH. Okay, this is glucose 6-phosphate and I am just isomerizing that into fructose 6-phosphate. So it will be looking like this. Both have the same number of hydrogen, carbon, oxygen, phosphorus, etc. Okay. And we know that CH2OPO3 2 minus H or OH. What will be the? Just tell. It will be H because OH is involved in this particular bonding. Okay, then here CH2OH OH H OH H HOH. So you can see the configuration difference, but both of them have the same number of carbon, oxygen. You can just count the number in both, it will be same only. If you are just converting glucose to fructose or fructose to glucose, the same isomerase enzyme only that will be catalyzing. And you can find one mutase enzyme in case of the glycolysis, that is also because which is converting 3-phosphoglyceraldehyde into 2-phosphoglyceraldehyde. 2-phosphoglycerate into 3-phosphoglycerate. That conversion is happening with the help of mutase enzyme, and that is also an example for the class isomerase. Okay, hope you understood what I mean. So that is what actually mean by the class of enzyme isomerase. Now let us move to the last class, that is Ligase. Ligase. So sixth class is Ligase enzyme. This you are familiar because you are learning this particular enzyme in DNA. When replication of DNA is there is Okazaki fragments, discontinuous strand that is joined with the help of enzyme ligase. So you have already learned in grade 12. So the same enzyme only. So ligase is actually what? The joining enzyme. Okay. So the one which actually link the two molecules. Okay. So that is what actually ligase. Link two molecules. And the best example is nothing but the DNA ligase. There are a lot of examples. We will be just covering that also when we are just discussing the metabolism. Otherwise, it will be beyond the scope of this particular enzyme classes. Okay, so DNA ligase is the best example for ligase enzyme, and ligase enzyme catalyzes the reaction where two molecules are linked to form a different one, to form a polymer. Okay, so that is what actually mean by the ligase. You can see in case of this particular thing what the bond between, so the phosphodiester bond between nucleotide is formed by the catalysis of ligase enzyme. So what will be ligase doing? DNA ligase create or form phosphodiester bond. Between nucleotides in DNA. Especially you can see that in case of Okazaki fragment, Okay, so that's about the ligase enzyme. So don't forget that. Hope you understood what is actually meant by the enzyme classes, what is enzyme, and we have already covered with the good examples that you will be repeatedly studying in metabolism part. So hope it's helpful for you and you understood the concept. Make sure that you are learning it properly before metabolism part. Okay, then it will be easy for you to understand the metabolism and how the reactions are happening, what kind of mechanism is working behind each reactions in metabolism. Okay, so for the convenience to upload in the YouTube, I have made this particular session in two. The first part will be dealing with the first 3 classes and second part is dealing with the second 3 classes. So total 6 classes of enzyme. So don't forget to start from the first part, part A, and then to the second part, part B. Okay, so I hope it's helpful for you and thank you for watching.